All right, let's dive into the vast, mysterious territory that makes up a whopping 98% of our genetic code. For so long, it was just dismissed as junk, but we're finding out it holds the keys to who we are. So let's get into it and start decoding.
So you've probably heard of junk DNA, right? For decades, the thinking was, look, if a piece of DNA doesn't code for a protein, what's the point? But that always felt a little incomplete. Because what if that 98%, this huge dark matter of our genome, isn't junk at all? What if it's the operating system that's actually running the whole show?
It was this massive puzzle. We had the complete book of life, our entire genome sequence, but we could only really read about 2% of it, the parts we call genes. The rest was written in a language we just couldn't decipher. But now, we're finally learning that this so-called junk DNA is absolutely essential. It plays a central role in, well, everything, from our evolution to our health and diseases.
Okay, so how in the world did we start to crack this code? Well, the first big clues didn't come from the DNA letters themselves, but from all these little chemical marks that were attached to the DNA. Turns out, these were the hidden messages scattered all throughout our genome.
Scientists call these epigenomic marks. You can basically think of them as chemical tags, things like histone modifications, that get stuck onto our DNA. And the key thing here is they don't actually change the A's, T's, C's, and G's. Instead, they act like this giant complex system of light switches, telling genes when to turn on and when to turn off in different cells. Here's a really great way to picture it. Imagine your genome is a giant, complicated instruction manual. These epigenomic marks are basically all the highlighters, the sticky notes, and the little underlines that someone left in the margins. They're telling the cell which parts are really important, which ones to skip, and which pages to read right now. Now, in the early days, researchers could only study one of these marks at a time. It was kind of like trying to understand a massive book by finding just a single highlighted word. But the real meaning isn't in one word, it's in the patterns. The huge new challenge was figuring out how to see all the highlights and all the sticky notes at the same time and understand what all those different combinations really meant. It was like trying to read an entire library at once.
This data overload, it required a completely new kind of tool. Something that could not only see all of these marks at once, but also make sense of their incredibly complex patterns.
And that tool is called ChromHMM. It's this really clever computational tool that was designed specifically to interpret these vast mountains of epigenomic data. It was built to see the big picture, to finally find the hidden grammar in all those genetic annotations.
And the big idea behind ChromHMM, the real game changer, was a fundamental shift in how to look at the data. Instead of focusing on individual marks in isolation, it was built to find their recurring combinations. Think about it like a language, right? A single letter, like T, doesn't mean much on its own. But when you see T, H, E together, you get a word with meaning. ChromHMM was designed to find the words of our epigenome.
So, how does this incredible mapmaker actually work? How does it take this unbelievably complex data and turn it into something a person can actually understand? Let's peek under the hood.
The whole process is actually pretty brilliant. First, ChromHMM just chops the entire genome into these small, manageable 200 base pair chunks. Then, for every single one of those chunks, it goes through and checks, is this mark here? Is that one? Yes or no? Next, it uses machine learning to scan across millions of these chunks to find the combinations of marks that show up together over and over again. And finally, it gives each of those recurring patterns a name, which creates what we call a chromatin state.
And that brings us to the key output, a chromatin state. What is that? Well, you can think of it as a functional label. It's a recurring signature of marks that tells us what that specific piece of DNA is probably doing. Is it a promoter, helping turn a gene on? Is it an enhancer, cranking up its activity? Or is it a repressed region, being kept quiet?
For example, if a segment of DNA consistently has, let's say, mark A and mark B present, the software might learn to label that an active promoter. But if it only sees mark C over and over again, it'll get labeled as a repressed region. It's these unique combinations that let scientists systematically map out the functions of the entire non-coding genome. And the end result of all this is a total transformation. You start with this chaotic mess of raw data from dozens of different experiments, and it becomes this simple, beautiful, color-coded map. You can literally look at a stretch of DNA and immediately see what it's doing. Red for active promoters, yellow for enhancers, gray for silent regions. It's a functional map of our genome. But look, this is way more than just making a cool-looking map. This is an incredibly powerful tool for understanding human health and disease, and this is where the science really starts to have a direct impact on all of our lives.
And the scale of this work is just huge. Get this. Using this exact method, big research projects have now mapped the chromatin states for over 100 different human cell and tissue types. That's an incredible library of genomic function.
Now, why is that such a big deal? Well, think about it. Every single cell in your body, from a brain cell to a skin cell, has the exact same DNA. So what makes them so different? These maps show us why. For the first time, we can see, at a really detailed level, which switches are being flipped on and off to make each type of cell unique.
And this brings us right to medicine. These chromatin maps are now being used to tackle some of our absolute biggest health challenges. Scientists are using them to understand what's going on behind the scenes in Alzheimer's disease, to pinpoint the regulatory switches involved in cancers and diabetes, and to finally make sense of all those genetic risk factors we find in big genetic studies.
And here is the crucial link. For years, we've known that many genetic risk factors for common diseases are located in that 98% of non-coding DNA. The problem was, we had no idea what they were actually doing. But now we can take those disease risk locations and lay them over these chromatin maps. We can see if a risk factor for Alzheimer's falls right in the middle of an active enhancer in brain cells. This finally lets us connect a genetic variation to a biological function and ultimately to a disease.
So just think about how far we've come. We went from dismissing 98% of our own DNA as junk to having these incredibly detailed functional maps that are helping us solve medical mysteries. We've learned how to read the notes in the margins of our own instruction manual. The only question left is, what other amazing stories are just waiting there for us to uncover?